Okay. Hey guys, this is a continuation of the special senses ear and eye presentation. We're going to pick up with the ear. Okay. All right. So initially, we're going to go through the just the structures of the ear, and then get into the mechanics of how the vestibular system actually works and how the cochlear portion works, and how we hear. Okay. So the ear, there's three different sections: external ear, middle ear, internal ear. Okay, we have starting from the outside here, we have the auricle or the pinna. This is the earlobe. Okay, and then we have external acoustic meatus. This is where the sound comes in. Okay, and here we have the ceruminous glands. Okay, this produces the earwax. Okay, and then the tympanic membrane. This is the divide from the external ear to the middle ear. The tympanic membrane, that's your eardrum. Okay, so we have the sound that comes in and it hits the eardrum and it vibrates that. And then it transmits that vibration through the middle ear and these auditory ossicles. Okay, these are tiny little bones in the middle ear. So there's three of them. Okay, the malleus, incus, and stapes are these little bones here. And then there are two little muscles you don't see on this picture. We'll see them in the next one, called the tensor tympani and the stapedius. Okay, those attach to the malleus and the stapes, and it restricts movement of these ossicles. So when we have a loud sound, we have a lot of vibration coming in on the tympanic membrane. There's a reflex. It tightens the tensor tympani and the stapedius, so it lessens the movement of these bones. Okay, so that loud noise doesn't cause these to over vibrate and cause any damage. Okay, so we have those in the middle ear. We also have, this is the tympanic cavity, okay, and that leads into the acoustic, the auditory tube here, the eustachian tube. Okay, so this is where this leads out into the nasopharynx. So this is how we equalize pressure between the middle ear. Okay, so when you're like on a plane and you're pulling out your ears or you're swallowing or chewing or something, this this tube opens up and it helps to equalize the pressure from the external ear to the middle ear. Okay, a lot of times I see clients with lip and tongue ties, and they have a soft palate that doesn't function properly. Okay, there's muscles on the soft palate when you swallow. It pulls on this eustachian tube and it opens. And so it helps to equalize pressure or drain fluid. But when these kids who have lip and tongue ties, they don't have a fully functional soft palate. It's not raising to contract the muscles properly. It doesn't open this eustachian tube. So we see a lot of buildup of fluid in the middle ear, causing ear infections and things like that. So I spend a lot of time kind of loosening this area, draining the fluid and stuff like that. So, but that's what happens here is when this opens, when you swallow, it equalizes pressure, or if there's some type of fluid that's built up here, it helps it drain out into the nasopharynx. Okay, so that's the middle ear. The middle ear ends when the stapes hits the oval window. Okay, and we'll talk about the oval window a little bit more. The oval window, that is the point at which the middle ear becomes the inner ear, and the internal ear is made up of semicircular canals and the vestibule. That's the vestibular system. Okay, and then the cochlear portion of it is this cochlea. Okay, this is where we hear. And then we see the vestibiocochlear nerve, facial nerve, okay, that comes in here. It innervates, the facial nerve innervates the stapedius. Okay, the tensor tympani is innervated by the cranial nerve 5, trigeminal. Okay, so here's a close-up of the middle ear. Okay, so we can see the malleus, incus, and stapes. Malleus, incus, stapes. All right, and then we see the muscles here. Okay, the stapedius muscle attaches into the stapes. Okay, and then the tensor tympani here attaches into the malleus. Okay, so it restricts, it actually tenses up the eardrum here and restricts vibration of the malleus there. Okay, so these muscles tense and restrict movement of these ossicles when there's loud noise. Okay. Then we move to the inner ear again. Okay, inner ear. So this is our vestibular cochlear system. Okay, so the vestibule is here. Okay, vestibule is here. It has the utricle and saccule. We'll talk about that in a minute. And then it has the semicircular canals here, the anterior, lateral, and posterior. And then we have the cochlear portion, this shell-like structure here. Okay, and we'll talk about how the sound comes in and stimulates hair cells so that we can hear. All right, so we have the bony labyrinth around it. Okay, it's covered in bone. So you'll see on the models in lab or the pictures of the models of the lab that it's covered in bone, and then you have a membranous labyrinth underneath it. Okay, so we have cross section cut out from here. We see that the bony labyrinth surrounds it all, and then underneath that we have perilymph. Okay, it's a fluid, and then inside of that, this blue portion is the endolymph. Okay, and the membranous labyrinth is the separation between the endolymph and the perilymph here. Okay, and we'll look more at that in a bit. Okay. So we're going to talk about the vestibular system first and equilibrium. So equilibrium is a state of physical balance. So this helps us to balance. So the semicircular canals or the semicircular ducts. Okay, there's three: anterior, posterior, and lateral. Sorry, these are blurry pictures. They're from your book. There's less blurry in your book. So the semicircular ducts, these monitor rotation. Okay, so there's three different rotations. So when you do the yes movement, when you nod yes, okay, that anterior rotation is monitored by the anterior canal, anterior semicircular duct. Okay, and then when we shake our heads no, when we go side to side no, that is measured by the lateral semicircular duct. Okay, and then the posterior one is when we do lateral flexion. Okay, so you can think of, usually I think of like a dog kind of looks at you and it doesn't know what you're doing and kind of gives you a questioning look. It kind of tilts its head to the side. So that's what the posterior canal monitors there. So it monitors those different rotations of your head. And then there are two sections that monitor, one that monitors horizontal movement and then one that monitors vertical movement. Okay, so that is monitored with the macula of the saccule and the utricle. Okay, so the utricle is sensitive to horizontal movement. So this is like if you're walking, just linear movement, like you're walking. Vertical movement is like you're jumping straight up and down. Okay, and that's monitored by the saccule. Okay, so we have horizontal movement, vertical movement, and then all the rotations taken care of by this vestibular section. Okay, so in the ampulla, okay, in the ampulla, the section is called the ampulla of each of the canals. We have something called the copula, okay, and a section called the crista. Okay, the crista has the hair cells, and then the copula is this gelatinous structure here that extends all the way through the edges of the ampulla. Okay, so this is within the endolymph. You see this is the blue portion. Okay, this is within the endolymph of the membranous portion of the vestibular system. Okay, so it's in that blue portion. So this is endolymph. Okay, so when we have movement of this endolymph, when we rotate through one of our rotations, okay, this moves this gelatinous structure, this copula, okay, one way or the other. Okay, and this movement then moves the hairs, the cilia, on the hair cell. Okay, these are the hair cell, and so we have stereocilia here, and the movement of this copula either moves the stereocilia towards the kinocilia or away from the kinocilia. Okay, this thicker one here is the kinocilia. So when it moves these stereocilia towards the kinocilia, we stimulate the hair cell. Okay, so we activate that. Oops, sorry about that. And then when we move away, okay, in this direction, away from the kinocilia, we inhibit the hair cell. Okay, so it doesn't activate the sensory nerve. Okay, So this can stimulate one way or the other, and so it allows us to tell which way our head is rotating. Okay. And then we look at, so that's in the ampulla of each of the semicircular ducts, and we have the crest, or the crista, and the copula, okay, and the hair cells that help us to determine rotation. And then we have the macula in the utricle and the saccule. And this is like an oval structure of hair cells. And it has a similar gelatinous material, okay? Similar gelatinous material, but then you also have these otoliths, okay, or ear stones, okay, on top of this otolithic membrane. So this gelatinous otolithic membrane, just like the copula, surrounds these hair cells, okay, the cilia of the hair cells. So when we move our head forward, when we're walking forward or backwards or jumping up and down, and we stimulate vertical or horizontal movements, okay, this gelatinous material, this membrane, as well as the otoliths, they shift. Okay, they shift one way or the other depending on how we're moving. Okay, and just like with the other hair cells, if we're going towards the kinocilia, okay, we're going to stimulate it. If we're moving the stereocilia away from the kinocilia, then we're going to inhibit that hair cell. Okay, so it tells us if we're moving forward or back. I don't like this picture because it shows rotation, which is not what this measures. So it just shows you how gravity kind of pulls at the otolith here, although it does not monitor rotation. So I don't know why they had that like that. But so these otoliths can actually break loose. So when you have vertigo, okay, sometimes these will break loose, okay, and it'll end up stimulating the hair cells in one of these semicircular canals. So they're overstimulated, so you end up feeling like you're falling or rotating one way or the other. And so you go through a maneuver called Epley's maneuver, which then you rotate the head certain ways and you sit up, and it's supposed to work the stone through the canal and get it out again, so it can stop stimulating those hair cells. But it's pretty effective for something called BPPV. It's just a vertigo that's caused by these otoliths. But if you have vertigo, then Epley's maneuver. Pretty effective. So that's how we sense our rotation and head movement, okay, vertical or horizontal as well. So when we have that information stimulating the vestibular nerve, okay, and we have the nerve that goes to the vestibular nucleus, okay, the vestibular nucleus then sends information all over the place. Okay, so we go to the cerebellum, okay, so we have that equilibrium from the semicircular canals and the vestibule, helping integrate with the joint mechanoreceptors and our visual stimulation to help tell where we are in space. Okay? And then we have down the vestibulospinal tract for postural muscle activation. And we have to cranial nerve 11, which we already know about. That is innervating the SCM muscle as well as the upper trap. So when we hear something, we can have that reflex where we turn our head towards the sound, and we use those muscles, SCM and upper trap, to turn our head. So that helps with that reflex. We go to cranial nerves 3, 4, and 6, which we know go to the eye, so eye movement. So the vestibulo-ocular reflex, when we turn our head, say to the left, our eyes, we can keep moving to the right to maintain our gaze on whatever it is. So if we're looking at the computer screen and we rotate our head to the left, then our eyes stay on the computer screen. So our eyes are moving to the right while our head moves to the left. And that is, so this is important in maintaining vestibulo-ocular reflex. And then we have fibers that go up to the cerebral cortex. So we have conscious perception of our movements. Okay, so it kind of goes all over the place. Now we're going to talk about sound, how we hear. Okay, so here's some just vocab words here for you for how we measure what we're hearing. So we have sound consists of waves of pressure of the air or of water, and so there's different frequencies. So the number of waves per that pass at a given point per time. Okay, so the higher frequency is a higher sound. Okay, and a higher pitch. The pitch is the sensory response to the frequency of the sound. Okay, so more frequency, more wavelengths passing that given point is higher pitch. The amplitude is the height of the sound wave, and the intensity is the energy the sound wave produces, the decibels. So let's talk about the cochlear system and the organ of Corti. Okay, the organ of Corti we'll see on the next slide better here. This is the organ of Corti. So the cochlear duct we see is here, this shell-like structure. Okay, so we have the cochlea here. We have the oval window here. This is where the stapes is. So we have the oval window. So when this vibrates at the oval window, it sends that vibration, that pressure wave through the perilymph here. Okay, remember the blue is the endolymph. And the perilymph is here. So this is the vestibular duct. Vestibular duct is the purple. Goes down the vestibular duct, goes all the way around, up here, and then goes to the, is that orange? And once it switches to orange, then that's the tympanic duct. Okay, and it comes back around all the way out and out through the round window. Okay, so in through the oval window, out through the round window. Okay, that pressure wave goes up and then back down. So we have the vestibular duct and the tympanic duct, and then this blue part is the cochlear duct with the perilymph in it. Okay, so when we look at a cross section here, when we slice it in half, okay, this is, we see the two different tubes. The vestibular system, or the vestibular duct, is always on top here. As we get from the oval window here. Round window is here, so we go up and around. The one on top of the organ of Corti, this is the vestibular duct, vestibular duct, vestibular duct here. And the tympanic duct is always on the inferior surface here. And that's the tympanic duct here, tympanic duct here, tympanic duct. Okay. So let's look at this one. It gives us a better view of the organ of Corti. Okay, this whole thing is the organ of Corti. Okay, there's some models of this in lab, so you're going to have to know the different parts of it. Okay, so again, the one on the superior side here is the vestibular duct. Tympanic duct is on the inferior side here. And we have the organ of Corti right here. So this is the basilar membrane. We have the hair cells here, and then this tectorial membrane, and that's within this cochlear duct, where the perilymph is here. Or I'm sorry, the endolymph. Endolymph is in this cochlear duct. Perilymph is in the vestibular duct and tympanic duct. Okay, so when we hear, this sends a signal. We have our sound wave, hits this tympanic membrane, hits our eardrum, it vibrates it. So it vibrates these bones, auditory ossicles, vibrates the oval window, sends this pressure wave through the perilymph. And then based on the frequency that goes through the cochlear duct at a certain point and displaces that basilar membrane, causing the hair cells to bump up into that tectorial membrane. Okay, so that's here. So we have the sound wave coming through, and it goes through the cochlear duct, and it displaces this basilar membrane. So it pushes these hair cells up into this tectorial membrane. When it pushes up into this tectorial membrane, it stimulates these hair cells, and that fires the nerves here. So the sound wave comes in, vibrates the tympanic membrane, vibrates the auditory ossicles, vibrates at the oval window, sending a pressure wave through the perilymph. And wherever that frequency hits and allows it to cross through the cochlear duct, displacing the basilar membrane. The basilar membrane then pushes the hair cells up against the tectorial membrane, and that stimulates the nerve. Then that pressure wave then travels back through the tympanic duct to the round window, okay? In through the vestibular duct, crossing the cochlear duct, out through the tympanic duct and the round window. So there's a couple of videos from the links I posted. I think I saw at least one, maybe two different ones on hearing, how we specifically hear that this whole mechanism walked through. So you may want to look at that and try to find that on one of the YouTube links that are up on the site. So once we have our stimulation of the cochlear branch of the vestibular cochlear nerve, this goes down to the cochlear nucleus. Cochlear nucleus then sends signals to the superior olivary nucleus. And those send tracks up to the inferior colliculus. And this is our auditory reflex center. Remember this, the superior colliculus has the visual reflex, and the auditory reflexes are from the inferior colliculus. And when we have fibers that go up and synapse here at the medial nucleus of the thalamus, and then out towards the auditory cortex in the temporal lobe. Okay, so that's the pathway that we see from the cochlear portion. And so that wraps up the ear. Okay, and so that covers both the eye and the ear. The last presentation in this one covers both the eye and the ear. So go over those these two and check out the links, because there's a few links on visual field as well as the events involved in hearing. So there's a couple of videos that can walk you through some of this special sensory of eye and ear. And so the next videos we'll do are for the other special senses, for taste and olfaction. Okay. Escape that. Stop sharing.